I would like to begin by thanking the organizers at LabRoots for the opportunity to present some of the work that we've been doing in my lab here at Microbiology Virtual Week. So to begin, my name is Maudry Laurent-Rolle. I am an assistant professor in the internal medicine department in the section of infectious diseases at Yale University. And today I will tell you about my work with CMPK2, and the title of my talk is CMPK2 Restricts the Replication of Multiple Flaviviruses. So the primary goal of my research program is to understand how host responses to viruses prevent or enhance disease. One approach to accomplishing this is by understanding how interferon-stimulated genes, such as CMPK2, which I will tell you about today, function and coordinate with cellular networks during viral infections. Hence, our overarching goal in the lab to characterize CMPK2 and define the molecular mechanisms by which CMPK2 inhibits virus replication, to identify and characterize host factors that interact with CMPK2 and either enhance or decrease its function. There is great interest in targeting interferon-stimulated genes for developing new antiviral therapies rather than targeting viral proteins in order to minimize emergence of antiviral drug resistance. Targeting an ISG like CMPK2 may be an effective strategy in the development of novel antiviral therapies. Upon infection, sensing of viral nucleic acids by the cytosolic pattern recognition receptors such as RIGI and MDA5 leads to the activation of downstream signaling factors, culminating in the activation of IRF3 and IRF7, which binds to the interferon beta promoter and induces expression. Interferon is secreted and signals in an autocrine or paracrine manner by binding to its receptor. Upon interferon binding to the type 1 interferon receptor, this leads to the activation of the JAK-STAT signaling pathway, resulting in the formation of the heterotrimeric complex, the interferon-stimulated gene factor 3 complex, which translocates into the nucleus and binds to the interferon-stimulated response element in the promoter region of interferon-stimulated genes such as CMPK2, which drives the transcription and expression of proteins, leading to an antiviral state to prevent viral replication and dissemination. Now, successful pathogenic viruses such as Flaviviruses and the SARS-CoV-2, for instance, have these viral antagonists that affect and inhibit these systems. So, for example, the Flaviviruses like Lung-Hat virus, Japanese encephalitis virus, West Nile virus, dengue virus, Zika virus have been shown from work that I did as a grad student that these viruses inhibit the interferon signaling pathway. And this has been work that has been done by myself and others. Recently, with the SARS-CoV-2, labs have shown that SARS-CoV-2 inhibits the production of interferon signaling as well as the interferon signaling pathway. So CMPK2 in humans is found adjacent to and inverted with respect to RS82, the ISG that encodes for the antiviral protein vibrin. This genomic organization is present in all vertebrates, and in lower organisms, CMPK2 and RS82 are fused. CMPK2 and viperin are co-transcribed upon induction of interferon signaling. CMPK2 encodes for the interferon-stimulated protein cytidylate monophosphate kinase 2, It's a 449 amino acid protein with a with a mitochondrial localization sequence at the N terminus, which is the first 24 amino acids. There is an N terminal domain and a C terminal domain. Now, the mitochondrial localization sequence causes CMPK2 to localize to the mitochondria primarily. Now, the first half of the protein, the N-terminal domain, has no known homology to any other protein, and the biological function is unknown. The C-terminal domain is conserved across species and is responsible for the kinase activity. CMPK2 was originally reported in 2008 in a publication in JBC to phosphorylate cytidine monophosphate and uridine monophosphate to their respective diphosphates using ATP as the donor. However, a recent report in 2018 from Stephen Alamo's group over at Albert Einstein, published in Nature, showed that CMPK2 principally operates on cytidine diphosphate and uridine diphosphate to form the triphosphorylated forms, cytidine triphosphate and uridine triphosphate, respectively. Now, CTP is then used as the substrate for fibrin, right? And fibrin then uses CTP to make DD8-TTP. DD8-TTP then gets incorporated into nascent RNA and acts as a chain terminator for RNA-dependent polymerases of Flaviviruses. After this paper was published in 2018, there was a paper from Karim's group in Nature that showed that CMPK2 plays a role in inflammation by activating the inflammasome. After this publication, there was a study in Science Advances that showed that CMPK2 plays a role in restricting HIV replication, but the mechanism by which CMPK2 inhibits HIV was not elucidated in that study. Afterwards, at that time, we started this project where we examined whether CMPK2 plays a role in inhibiting viral replication, and at the time we began that study, we used the Zika virus, a Flavivirus, as our model. So Zika virus is an RNA virus in the Flavivirus genus belonging to the Flaviviridae family. It's closely related to dengue and West Nile virus and yellow fever virus. Zika virus was actually first isolated in 1947 and remained in relative obscurity for about 70 years or so until 2015 when it was transmitted in Brazil, introduced to Brazil in 2015 in 2015. Now we know that Zika virus is transmitted in the human to mosquito human transmission cycle. However, there is evidence that shows that Zika virus can be transmitted from the mother to the fetus, and there are also reports of sexual transmission. 1 in 5 people who become infected become ill from Zika virus with symptoms of a rash, fever, and conjunctivitis, and symptoms last anywhere from 2 to 7 days. There are no treatments, and treatment is supportive, and there are no vaccines. Therefore, to assess the biological effects of CMPK2 in controlling Zika virus infection, we generated a wherein we can examine wherein CMPK2 can be regulated in a time and dose-dependent manner. So very briefly, full-length CMPK2 was cloned into a lentivirus response vector system controlled by a tetracycline response element. 0 cells were transduced with this lentivirus encoding either CMPK2 or an empty vector for our control. And the reason we use the 0 cells is because 0 cells has a genetic defect in producing interferon. However, the cells do respond to exogenous interferon. So this gives us the advantage of 1, being able to turn on the ISG of interest, in our case CMPK2, without turning on other interferon-stimulated genes, and 2, being able to examine the effects on viral replication by regulating expression of the protein of interest. So the first thing we did after we made the cell, we examined whether the CMPK2 is expressed upon treatment with doxycycline, and here in panel A, you can see the expression of CMPK2. In panel B, we examined by confocal microscopy the subcellular localization of CMPK2. As I said at the beginning, CMPK2 has a mitochondrial localization sequence that localizes it to primarily the mitochondria. And what you see in panel B in cells treated with doxycycline is the expression of CMPK2 in the second panel, the lower panel. And in the third panel, we have the mitotracker, which is the marker for the mitochondria. And what we see is co-localization of CMPK2 with the mitotracker, confirming that CMPK2 is primarily localized to the mitochondria. Next, we examine the effect of CMPK2 on Zika virus replication. We use the cells and we either mock treated or treated with doxycycline for 24 hours. So we allow the CMPK2 to be expressed, as I showed in panel A. Then 24 hours post-treatment with doxycycline, the cells were infected with Zika virus with a multiplicity of infection of 1. From now on, I will just say MOI, so an MOI of 1. 24 hours post-infection, we harvest the supernatant, and we examine for viral replication, quantification of the virus by plaque assay. And what we observe is that in cells treated with doxycycline and expressing CMPK2, that we had about a tenfold, which is a log difference of Zika virus replication. So in comparison to cells not expressing CMPK2. So that result showed us that CMPK2 has an effect on Zika virus replication. Next, to complement our CMPK2 inducible cell system, we use a CRISPR-Cas9 approach to deplete CMPK2 cells, CMPK2 in human phosphine fibroblast cells. So very briefly, a lentivirus vector was used to deliver either a non-targeting guide RNA, which is our control, or a guide RNA to targeting CMPK2. We then confirmed that CMPK2 was knocked out in the cell, and then we used a lenti CRISPR vector to deliver a guide RNA against Cas9, and that was done in order to guard against off-targeting effects. We then validated that our gene of interest was removed by, in panel A, by qPCR, and here you see a knockdown of the mRNA, right, the message, and by Western blotting. And here we see in wild-type cells and the non-targeted human phosphine fibroblast cells, treated with interferon, you have upregulation, again, of ISGs. So you see expression of the protein, CMPK2, and here, viperin is just a control. You see expression of viperin. And in the CMPK2 knockout cells, you don't see CMPK2. Next, we use these cells to evaluate the effect of Zika virus replication. So again, we use the cells to infect them with Zika virus at an MOI of 1. 6 hours post-infection, we treated the cells with mock treated or treated with interferon. This will allow the cells to, interferon will bind its receptor, right? And there will be upregulation of all these ISGs when you treat with interferon. 16 hours later, we harvested the supernatant for virus quantification by plaque assay. And what we observed is that in cells, the non-targeted cells, the empty cells, as well as the CMPK2 knockout cells, When these cells are not treated with interferon, you have similar virus titers. However, as expected, in the non-targeted cells, interferon reduces the replication of Zika virus in the cells, again because when you treat the cells with interferon, you have an upregulation of all these ISGs and you have a reduction in viral replication. However, in the CMPK2 knockout cells, there was no difference in replication between the non-interferon treatment versus the interferon treatment, which confirms that CMPK2 plays a role in Zika virus replication. Given the similarities in the molecular biology of the Flaviviruses, we examined whether CMPK2 plays a role in the replication of other Flaviviruses. So to do that, we use our doxycycline-inducible cell system, and we would mock treat or treat with doxycycline. 24 hours post-treatment, again, we would infect with our virus of interest. In this case, we're going to examine a few medically relevant flaviviruses, and then we would, 24 hours later, we would collect the supernatants for analysis by plaque assay. Now, in these studies that I will show you also, we will also have viruses expressing fluorescent proteins. And in the studies, when we have viruses expressing fluorescent proteins, we will harvest the cells and we will do flow cytometry for analysis. So what we saw is that CMPK2 inhibited dengue virus replication. CMPK2 inhibited pungent virus replication, another flavivirus. CMPK2 inhibited yellow fever virus, another flavivirus. So our results so far show us that CMPK2 inhibits multiple flaviviruses. Given the results that CMPK2 inhibits HIV replication, as I said, that was published sometime in 2018, and our results with 4 different flaviviruses, we ask whether CMPK2 may affect the replication of other virus families, including the Coronaviridae family, the Orthomyxoviridae family. And again, we use our doxycycline-inducible system for viruses containing a fluorescent protein. We will use FACS analysis for analysis, whereas if the virus does not express a fluorescent protein, we will use plaque assay. And what I'm going to show you here is that we examine CMPK2 effect on a virus of the coronavirus, infectious bronchitis virus, which is an avian virus. And what we see by plaque assay analysis is no difference, no effect on IgG replication. Next, we examined whether CMPK2 affected SARS-CoV-2, the etiologic agent of the current COVID-19 pandemic, and we saw no difference by plaque assay. Next, we examined whether CMPK2 affects other relevant viruses. So, for example, we looked at influenza virus. Here we're using an influenza virus expressing a fluorescent protein. This is TRGFP. And here, GFP positive cells. In cells expressing CMPK2 or not expressing CMPK2. We saw similar results with the Newcastle disease virus, which belongs to the Paramyxoviridae family. Similar results with vesiculostomatitis virus belonging to the Rhabdoviridae family. And we examined a DNA virus, herpes simplex virus, belonging to the herpes viridae family, and we saw no effect of CMPK2 on these viruses. So, given Given the effective antiviral activity of CMPK2 against multiple flaviviruses, including dengue, Zika, and yellow fever, CMPK2 actually would make an attractive target for the development of novel antiviral therapies. Now, dissecting the mechanism by which CMPK2 inhibits these flaviviruses may yield new strategies for translating the antiviral activity of CMPK2 into a clinically useful compound. So to look towards the mechanism of action, we first look towards the, the domains of CMPK2 and what are the requirements for these different domains. So to begin, we first ask the question, does the mitochondrial localization of CMPK2 is that required for its antiviral function? So again, as I've shown you before, that CMPK2 has a mitochondrial localization sequence that takes it into the mitochondria. You can see that in panel A, the lower part of panel A when treated with doxycycline. So for our studies, we generated this expression plasmid either expressed in an empty vector as a control, or wild type CMPK2 in panel B, or a variant lacking the first 22 amino acids. So one would expect that this variant would lack the ability to localize to the mitochondria. First thing we did, we examined for protein expression by immunoblot analysis and observed that both the wild type and this delta MLS, which lacked those first 22 amino acids, were expressed to similar levels. Next, we examined the subcellular localization by confocal microscopy, and we observed that the wild type, this is in panel B, upper part of the panel B, the wild type co-localizes with the mitotracker, which is the marker for the mitochondria. However, the delta MLS, which lacks the 22 amino acids required to take it into the mitochondria, remains cytosolic as expected. So next, we examine the effect of this mutant protein on Zika virus replication. So to do that, we used 293 T cells and we transfected them with these expression vectors. 24 hours post-transfection, we infected the cells with Zika virus at an MOI of 1. And 24 hours post-infection, we harvested the supernatant for quantification by plaque assay. And what we observed is that the wild type CMPK2 inhibits Zika virus replication, which confirms what we saw with our inducible system in the zero cell. However, the CMPK2 variant lacking the first 22 amino acids, which is no longer localized to the mitochondria, lacks this ability to inhibit Zika virus replication. To ensure that this is not an effect of expression, we use immunoblot analysis to look at the expression of these proteins, and we saw similar levels in panel B of the wild type and the mutant. We also examined the envelope protein, which is one of the structural proteins of Zika virus, and saw that there was a decrease in the envelope protein in the wild type cells as expected, as Zika virus inhibits the replication of CMPK2 inhibits the replication of Zika virus. So what this tells us is that the mitochondrial localization of CMPK2 is required for its antiviral function. Next, we examined the contribution of each of these domains. As I said at the beginning, you have the mitochondrial localization sequence, and you have the which is variable between species, and you have the C-terminal domain, which is well conserved. And the C-terminal domain is responsible for the kinase activity. And we generated these doxycycline-inducible cells expressing these mutant proteins for our studies. So we have the wild-type protein, the deletion mutant, which I've shown you before, would not inhibit Zika virus replication. We made a catalytically inactive CMPK2 protein, mutant protein, and we mutants expressed in the first half of the protein, so an N-terminal domain with and without the mitochondrial localization sequence. And we have the C-terminal domain with and without the mitochondrial localization sequence. So we first examine the expression of these mutant proteins by immunoblot analysis. And what we observe in panel B is that the mutants expressed in only the C-terminal domain, they are very poorly expressed. And moving forward with our experiments, we did not use the C-terminal domain. Next, we examine the subcellular localization of these mutant proteins. And as expected, the proteins containing the mitochondrial localization sequence, for example, the wild type, the catalytically inactive D330A, as well as the N-terminal domain 1 to 201, they are all localized primarily in the mitochondria. However, the mutants lacking the mitochondrial localization sequence are primarily cytosolic, like delta MLS, and the mutants 23 to 201. Then we examine the effect of these mutants on Zika virus replication. And what we observe is that, in keeping with what we've seen before, that the wild type reduces Zika virus replication, unlike the delta MLS, which I've shown you before, does not inhibit Zika virus replication. Now I'd like you to go to, so we're in panel B, and let's go to the last few bars. So that is the 1 to 201 to the 23 to 201. And what we see with the 1 to 201 is that this first half of the CMPK2 inhibits Zika virus replication, which means that the C-terminal domain and the kinase activity is not required for CMPK2 antiviral function. Here in the last part, you see here that the 23 to 201, this is the first half of CMPK2 lacking the mitochondrial localization sequence, is unable to inhibit Zika virus replication. Again, highlighting the importance of the localization of CMPK2 in the mitochondria for this antiviral function. Now, as I said, the C-terminal domain and the kinase domain is not required based on these data, and the catalytically inactive CMPK2, where an aspartic acid at amino acid position 330 was changed to an alanine. Yeah, this catalytically inactive CMPK2 is also able to inhibit Zika virus replication, confirming that the kinase activity of CMPK2 is not required for its antiviral function. So what I've shown you today, in conclusion, is that CMPK2 inhibits the replication of multiple Flaviviruses, making it a very attractive candidate for use as an antiviral. I've shown you that the mitochondrial localization of CMPK2 is required for its antiviral activity. And I've shown you that the N-terminal domain of CMPK2 and not the C-terminal domain, not the kinase activity, is required for its antiviral function. Now, this brings us to some of the more current experiments that we're doing and more current questions that we're asking. So CMPK2 has this N-terminal domain that is highly variable with no known function. And I just showed you that this N-terminal domain and the mitochondrial localization is required for its antiviral function and not the C-terminal domain and the kinase activity. I've shown you that the recent report a few months ago, 2 months ago or so, by Lei et al. That showed that CMPK2 inhibits dengue virus replication, along with the report in 2018 that showed that CMPK2 inhibits HIV replication. Makes it even more interesting, and hence why we're doing these current studies right now, trying to identify the specific amino acid residues within the N-terminal domain that is required for this activity. As I said, we have no idea what the first end terminal, the first half of the protein, what the function is. So this would be a first step in trying to understand what the implications of those results that we're seeing. Some of the other things that we're doing, we're trying to figure out what are the host factors that interact with CMPK2 and enhance or decrease its function. We have already used the biotin proximity labeling approach to identify cellular proteins that are proximal to CMPK2, and we are currently validating this hit using immunoprecipitation assays, gene knockout studies, to try to figure out what is the biological relevance of the hits that we found. Something else we have to figure out is what are some of the effects of CMPK2 on cellular metabolism, on nucleotide biosynthesis. And interestingly, what we're doing now is trying to figure out, will Zika virus develop adaptive mutations to antagonize CMPK2 function? So at the beginning, I showed you that slide that Flaviviruses have evolved to inhibit host immune responses to ensure their replication and survival. Persistent expression of interferon-stimulated genes like CMPK2 may result in selection pressures, and subsequently viral crisis species may emerge that can overcome the host immune response. So far, we have done serial passaging of Zika virus in cells expressing CMPK2. We have isolated virus from the supernatant and have sent this for sequencing. And currently, we are analyzing this data. So we have many interesting avenues that we are following for CMPK2. And with that, I will end here, and I would like to thank Joanna Pollack, who is a postdoctoral fellow who joined my group last October and has really taken this project and has really done tremendous work with it. So I am very grateful for her help and joining my lab this early on, as I only started my lab last July, actually, so about a year ago. And Jack, who has been working with Joe also and collaborating with us on this project, so I'm very grateful. And many thanks also to Jack for other projects that we've collaborated on, which we did not talk about here today. Maybe we'll come back next year and talk a little bit more about some of this work. I would like to thank the Cresswell Lab, Peter Cresswell, my mentor. I did my postdoctoral studies with Pete, and actually all this work, we started this work with Pete when I was a postdoc. And he has been very supportive, and I've learned so much from him, and I continue to learn a lot from Pete. And Patrick Hahn, who is a postdoc with Pete, who has done some studies with CMPK2, is helping us with some of our studies, and that I did not show today, and other members of the lab. I would like to thank Juliette Morrison and her lab for collaborating with us on the studies with dengue virus and CMPK2. And some of the labs here at Yale who have been very generous, helping me with these reagents, Errol Pickrick and his lab. And Errol is also my section chief in my department. So thanks for his support. Craig Weiling, who helped us set up all our Akiko and her lab, Adolfo Gasser-Sastre, who I did my thesis work with and who continues to be very supportive, and Lisa Miorin in his lab, who is now actually an assistant professor. Environmental health and safety here at Yale, who has been critical in helping us with setting up the SARS-CoV-2 work on some new projects that we're actually starting now. All the cartoons were done with BioRender, and I would like to thank our funding source, the Yale Center for Clinical Investigation, for their award for support. And Joanna is supported by the Laboratory Medicine. Thank the audience again. Thank you.